So let's start from question number two from 7.2, which is the population in Hardy Weinberg. Okay, so we have discussed question number one, which is related to cats already with the fur. We have discussed this in detail and we have already found the Hardy Weinberg theorem, how to apply the Hardy Weinberg equation on this. If I give you question, would you able to attempt it? Wait, then I'll give you the same question that we have already done. Let's see, would you able to understand the statement and would you able to um, know what equation do are we going to use for this one? Would it be 1 is equal to p plus q, or would it be like p square plus 2pq plus q square? Okay, Adiba, are you hearing me? So here is one question in a population. 36 of the cats have a F allele. So it was given in a question in the very starting statements that the F is the dominant allele and stops the formation of pigment in the fur that results in the white fur, while the small f has is recessive, has no effect on the fur color. Okay, so this one is the dominant allele. Clear? Now, in the question they have asked, use the Hardy-Weinberg equation to calculate the frequency of the small f allele and show your working. Okay, so it means first of all we need to write the equation that 1 is equal to, like, the both populations, the recessive one and the dominant both. Yes, it would be, but let's first focus on this. I want, so this is the general thing that we use and then further we take care. Is it so? Okay. Okay, so for example, if this one is the dominant one and this one is the recessive one, what would be the value of recessive? This is the, okay. P is the dominant one, Q is the recessive one, and you want to find out that if if the dominant alleles are 36%, what is the frequency of F alleles, this recessive one, in this population? So how you are going to find it out? 64%. Yes, it would be like Q is equal to 64%. Okay, wait. We haven't reached it yet. So, what we need to calculate the frequency of L allele in this population. So, when we talk about population or when we talk about the frequency, that would be equal to 1 is equal to P square plus 2PQ plus Q square. Let's say the P square is 36, that we already know. We don't need to know this. And this one is 64%. Is it so? So how you would find Q? If this is Q square, how would you find Q? What you would do? Yes, rearrange it and do what? And take a square root. Look, it's a Q square. Then you need to take a square root. Yeah. So what would be answer for 64%, which is 64 divided by 100? Point. Okay, 0.64. Take square root of 0.64. 0.8. Correct. Okay. So wait, let me just erase this all. Give me a minute. is clear. So we have a question from again question number 2, which we didn't discuss. So let's discuss this. That we have give two type of cells that can stimulate immune response. What are the two cells? Yes, immune response. Sorry? No, it's from the same paper. It's question number 2 from 7.2. What stimulate pathogens when they enter your body? Okay, if they're not pathogens, they can be some type of what? Some type of antigens. Yes, they can be. They can be cancer cells as well. So the autoimmune disease causes the immune system to attack the healthy body. That's why it is known as autoimmune. Scientists investigated the immune response of healthy mice and mice with autoimmune disease. A chemical OXA, this is a chemical that causes an immune response in mice and can make their skin swell. Mice had olive oil applied to their left ear and OXA in olive oil applied to the right ear. So the immune response was recorded in two ways. Number one, the cellular response by measuring the mean increases in the ear thickness, and the second one is the humoral response by measuring the concentration of anti-OXA, means the concentration of antibody. So one is after 24 hours and the other is after 14 days. Why we cannot calculate the antibody concentration in the body right after the infection, like after 24 hours? Why we are taking 14 days over here instead of 24 hours? First of all, the concentration of antigens increases dramatically, and then it decreases. So there is no production of antibody in the meanwhile. Why so? There is no antibody. Why? Why this antigen has increased to a lot and there is no antibody production in the first 3 days? Because our body is preparing the right antibody for that right antigen. You can say it's just like a trial method. Our body is doing a trial method. Okay, we can maybe this antigen can be killed with this type of antibody. So the our body or our immune response is putting the correct sequence in the antibody, because antibody is a protein. So maybe in those 3 to 4 days our body is preparing the right antibody for that specific antigen. Okay, so this is the reason that why we have taken it after 14 days, because after 14 days the amount would be you can say the sufficient to calculate or to compare it with some other species. Okay. Okay, then they have given us a table and it gives us the values in the standard deviation as well, and it is for about 95% of the data. Why do you think the olive oil was applied to the left ear of the mice as a control? And how would you define a control? Okay, show that OXA is controlling the autoimmune, the autoimmune response. Okay, because we are discussing autoimmune response. Let's move to the next question. Put a suitable graph for mean increase in the ear thickness for each group of the mice in table 1. So we have to use this one table, okay? And then, are you getting me? And then we have, first of all, we need to give it like a proper x-axis, the labeling, and then for what one big box or one small box is equal to what value. First of all, what would be on x-axis and y-axis? How would you label it? So the graph is between, let's read the heading. The graph is between the mean increase in the ear thickness. So how would you suggest that we would draw a graph? Would it be like a line? Wait, let me ask something. Would it be a line graph, a bar graph, or a like a pie chart? No, they have given this, so we can have two options, line or bar. So which graph would you are going to choose? Bar graph. Bar graph is used. Okay, we use line graph when on one of the axes, or like we what we have to tell only the mean increase. Okay, let's say mean increase in the ear thickness. Okay, we they haven't given us any other value to write on the other axis. For example, if on y-axis we are writing this mean increase in the ear thickness, they haven't given us anything to write on x-axis. Okay, for example, if they have given us days like 1 day, 2 day, 3 day, 4 day, 5 day, then we can write here for each day that day 1 value is this, day 2 value is this, day 3 value is this. Only then we can use line graph. But here they have given us only 4 values on the very same day, which is 24 hour after 24 hours. So we need to use a bar graph. Again, I'm telling when to use line graph or when to use bar graph. Bar graph is used whenever they haven't given us the values over a period of time. If they have given us only one value for any given time, for example 24 hour, or for example 7 days, they haven't given us value for the days in between or for the hours in between, then we have to use bar graph. But if they give value of all the hours, like 1 hour, 2 hour, 3 hour, 4 hour till 24 hour, only then we can use line graph. Clear? Okay, so on y-axis we are going to write the mean increase in ear thickness. And what's the unit? So we have to mention the unit in graph. Let's go back and check the units. What are the units? It's per centimeter into 10 raised to power 3. Okay, so let's write per centimeter into 10 raised to power minus 3. So this is what you need to write on y-axis. Everything. Okay, now how to select boxes? Let's see how many boxes are there. So these are like 1 box, 2 box, 3 box, 4 box, 5 box, 6 box, 7 box, and 8 box. Okay, so we have 8 boxes, and the maximum value that we have is 25 or 26. So what would be the one box value? One big box is equal to what number you want to choose. We have 8, 3, 4. What number do you want to choose? 3 for one big box or 4 for one big box? I guess 2 would be won't be feasible, because the two, if you select 2 for one big box, let me tell you. For example, you're starting here with 0. Let's write here 2, 4. I mean this, like this whole box. So if we choose 2, the reading would be till 16, not more than that. But we need a reading till 26. So what do you want to choose? 4, okay. 4 is easy to determine as compare, or you we can also use 3 as well, but 3 again not working. If we choose 3, it would be like 24. So let's say 4. So it would be 4. This one big box would be equal to 4. This would be equal to 8, 12, 16, then we have 20, 24, and then 28, okay, and then 32. Clear? Now, we need to choose all these four, the healthy one and the autoimmune one. Let's label these. We need to make two bar graphs, let's say. Maybe my line won't be straight. Just give me a minute. It will be like one bar here. These bars should not be touching. One very important thing. So maybe second bar, starting from here, third bar, and fourth bar. Okay? Then we need to label the bars. So we have four labelings. We have female healthy, male healthy. We have autoimmune disease male, and we have autoimmune disease female. So we will name it healthy male, healthy female. Are you getting me? Then we have autoimmune. I'm writing IA for right now, but you need to write the full word. Autoimmune male and autoimmune female. Okay? So we need to write the heading. Now, whatever the value, we choose that value and then write it. Like for if it is 17.9, so it must be around here. If it's 16, it must be around here. So we need to make a proper bar. Let me see if I can make. Yes, I do have an option. So it would be from here. Let me make it bigger. So it must be like this. Okay? This is how you make a bar graph. Now the important what important things have I told you while making a bar graph? Oh, any graph, yes. What you need to keep in mind while making a graph. Okay. First of all, to label the axis, then to choose the then to choose the right value or for each box, and then what other important thing that you need to keep in mind while making a bar graph, that these bars should not be touching each other. Okay. Okay, let's move to the next point. What can you conclude about the effects of autoimmune disease on the cellular response and the humoral response in male and female mice? So here they have given us the mean values, so of the antibody concentration. So what can you tell this from, like, what can be the cellular response and what can be the humoral response? Okay, what about standard deviation? Let's move to that. Yes. How you are saying that there is no significant difference by looking at the standard deviation, right? Okay, exactly. If they overlap, then we can say that if we look at the female, as there is more overlapping in the female. S- so Okay. There is yes, there is less increase in the response. Okay, we can say that. And what about the humoral response? So we say it's increasing from 14 to 26. That's what antibody is. If we look at 16 to 14, 16 and 14, they're quite closer to each other. So there is an overlapping. Look, it's 16, let's say. It's 16 and it's 14. Let's do plus minus plus minus. These values are overlapping for males, so there is no significant difference in this one in the male to male response. Okay, but in female there is no overlapping. It's 14 and it's 26. 26 minus 7. It would be 19, right? It would be 19. 26 minus 7. The higher value for this one is 18. So there is no overlapping in this one. So we can say that there is a significant increase in female to female humoral response. Okay? Have you got this point? How do we calculate? Okay. Next question. Some studies have shown in the human, estrogen has the opposite effect on two different autoimmune disease. Estrogen accelerates the progression of SLE and prevents the progression of RA. And the table is given. What is the effect on of estrogen on humoral response and cellular response? So in healthy mice there is no effect, but in the autoimmune disease or the disease mice, the humoral response increases while the cellular response decreases. Okay, this is what we get from table. Okay, wait. Whatever you're saying is correct. I'm just asking, you need to evaluate the student's conclusion. Like, what is the conclusion of student from this table? What has student included? Okay, let's what is the effect of estrogen on the humoral response? Let's see this. Okay, more antibodies. Okay, it decreases the cellular response. Okay. Okay, correct. So this is the thing that you need to write for 4 marks. 2 points for humor response and 2 points for cellular response. And give one reason, okay? If the humor response is increasing, this can be the reason that it's increasing. And if the cellular response is decreasing, so this is the reason why it's decreasing. Clear?